I didn't see you there. We will be doing a lab today to show the process of transpiration, or at least kind of sort of part of it. We have dissected seeds, so you can see the cotyledon gives nutrients to the little embryo plant so that it can start to grow. And the plant, the seed and seedling need water, right? We kind of sort of know that from what, first grade, where we know that the roots bring in water and supply water to the plant, but how do they do that? So if we look at the plant and compare it to us, we know that when we learned about microorganisms and multicellular organisms compared to single-celled organisms, the single-celled organisms have the characteristics of life. They don't need help from other cells, but multicellular organisms need other cells to function. Our cells make up tissues, and those tissues make up organs. In plants, they have tissues and organs as well. So I'm going to compare some of them. The roots, it's kind of like being able to take in water. So like us, it's our mouth and we take in water. Imagine that their roots are little mouths for nutrients and water. They also they don't have lungs, but they need to take in gases. We take in oxygen and we release carbon dioxide. The plant takes in the carbon dioxide and releases oxygen. What we're going to show today is how water travels from the roots through the stems to the leaves. When we get into learning more about the leaves, we'll talk about a little bit more the next step. So what we're going to use today is celery. Cool thing about this, if you've ever seen an actual celery stalk celery stalks growing out of the ground, this part here that's cut off, there's a bunch of little fibrous roots coming off, and that's what takes in the water. But just like if you get a flower, you go pick a flower and it's got a stem and you put it in some water, it'll stay alive for a little while before it dies. And that's because there's still water able to come up and keep it alive for just a little bit longer. It doesn't function as well as roots though. So we're going to do that. And what we're going to be looking for are the little tubes that are from the roots all the way through the stems and go up through the leaves. Just like in us, we have a circulatory system, so we have arteries and veins that carry blood that has oxygen and nutrients to all of the cells in our body. That's the same thing that these little arteries and veins, but they're not called that. They're called xylem and phloem. Xylem is the stuff that's in the little tubes that go up from the roots all the way up to the leaves in every single part of the plant, and that brings things up. Phloem takes the sugars that are produced in the leaves through photosynthesis down to everything else in the plant. So with our celery, first thing I'm going to do is make a nice clean cut. So that's the first thing with my fancy science tool. I want to, first of all, find the ones that have lots of leaves on them. So it's kind of hard to find these in the store because most of the time they cut those off because people don't want the leaves. Oh, this is a good one. Okay, lots of leaves on that. So I'm gonna pick a few out, and this is really easy to set up. That's a good one. Okay, make sure we've got a nice clean cut, and then we're gonna put them in water that has food coloring in it. The idea here is that if this water actually does what I told you it does, if it actually comes up through the stem and out the leaves, it's going to change color because of the food coloring in it. So if we check on this in 24 hours, 48 hours, we should notice some different colors in here. That's the idea. There is no reason why I have 3 different colors of water except for that it's cool, right? We'll be able to see what happens and change the colors of these. So maybe we'll have, I don't have enough food coloring to make a rainbow, but that would be cool too. I'm going to stick, let's see, I'll find a few more with good leaves. This one too. Let's cut these up. I'll put 2 celery stalks with leaves in each of the jars, and we will check again in a day or 2. I'll let them sit for a while and see what happens. And then the fun part of this is we're going to get to look at the internal structures, the xylem and the phloem, with a microscope and see what it looks like with that as well. All right, so yeah.
Now, the calculator without keys. Sharp, super thin man, world's thinnest, approximately 6/32 of an inch. Its electronic tone verifies your entries, and its miracle energy-saving circuitry can provide years of battery life. Automatic shutoff avoids battery drain. The super thin man, complete with batteries and wallet, from Sharp, the calculator specialist.
48 hours later, these celery stalks have been in the different colored water, and they are now different colors. It's hard to see the green and the blue, but I guess if you're real close, you can see the difference at the very ends of the leaves. Yeah. The red kind of looks more brown than red, but we'll be able to use these and we'll see what happens. So the purpose, remember, of this lab is to see how transpiration works, that the xylem are the little tubes that come up through the plant and take water in from the roots through the xylem and all the way out to each cell. So each leaf has had the water coming up and out of it, and then it evaporates out of the leaves. This is transpiration, right? The movement of the water through the plant, in from the roots, through the shoots and stems, all the branches going out to the leaves. When we look at these in the microscopes, we're looking for the xylem to be the color of whatever water it was in, because it's been stained as the water moves up through it. We should be able to see the same thing in the leaves. The easiest one to see is probably going to be the red, so that's the one I'm going to start with. In class, I have this little razor blade that I use to get really, really thin slices, so I don't know how talented I am with this great big giant knife. I'll try to, there we go, try to get some really thin slices. For the leaves, we'll just take a little section of each. And when we prepare the slide, we'll put a little bit of water on it and put a cover slip on it so that we can see it nice and close and flat. Hopefully, you now understand how the xylem works. It's been stained as the water moved up through it. The phloem, can't really see it because the water is not going through it. It's the sugars created from photosynthesis in the leaves that flow through the rest of the plant in the phloem. Phloem, get it? This is a bunch of celery cells. This is one of the stalks that was in the water with red food coloring, so we're looking for red. We'll just slide this up. There we go. And that is xylem, where the water came up and stained that red. Let me get another one here. This is, again, a bunch of celery cells, pretty green celery cells, and there we go. And there's the xylem. So xylem is a hollow tube. It's just made up of dead cells. It's a hollow tube. The water moves up through the tube, and since the water had red food coloring in it, it was stained red as it was moving up through it and out the leaves. I want to show you a still picture because what happens here is that the camera I'm using, even though I magnified it 100 times, it's actually showing it much bigger because the camera can't take a circle and its image is a rectangle instead. So it actually is appearing much larger than it would if you were looking through it. So let's go ahead and get a still shot of one so you can see what it actually looks like. Not quite so close. So the xylem is what was stained red. All of these little red spots here are the tubes carrying the water up from the roots. Then the phloem, it's the tubes next to the xylem that are not stained because there was no water moving up through them. You notice how it's not coming off the paper towel easily? Those roots are clinging to it. And that's what they do to the ground. The roots cling to the ground. Look at how big that is. You see that?
Mm-hmm.
The roots cling to the ground, and that's one purpose of the roots, is to anchor it into the ground. So we're going to look at this in the microscope. We're going to look at the little structures in the roots in the microscope. This is the very, very end of the root from the radish seed. So that's the very end of it. And this is inside of it. You can see all of the cells. It's pretty cool. And again, this microscope, using this camera, it's hard to get a view of from far enough back. So this isn't quite as exciting as I would like it to be. So let's try a different one. This one is a root from the rye seeds. This is grass. And if you've ever pulled grass out, if you've ever tried to pull one blade of grass out, it's got like one long root, or it looks like it is, but look at this. There's little teeny tiny hair-like projections coming out that help hold it into place. Let me see if we can get the focus here. There they are. Look at that. Isn't that cool? So they are all the way around the root, and they're helping to anchor it into the ground. Super cool. The next video will be about leaves and photosynthesis, but since we had that celery soaking in the water with red food coloring, I thought I'd go ahead and show you these red lines in the leaves. These are the little veins in the leaves, meaning that all the way into the leaves, we have xylem. So it's going all the way from the roots through the shoots and the stems to the leaves. All right, let's go up to 100 times magnification. There we go. Look at that. Isn't that cool? So this is the end of the road. What you can't see in this, and what we're going to be looking at in the next video, are the structures where the water is released from the end of the xylem. So stay tuned. Remember the water cycle? It starts with the sun. The sun heats the earth and the water, and then the water turns to vapor and evaporates. As it rises up into the sky and begins to cool, it condenses. When the clouds get too heavy, it falls as precipitation. As it's falling, it might land back in the water, it might land on the ground. If it's on the ground on a hill and goes off into a lower area, that is runoff. If it sinks into the ground, it's groundwater. Some of the water that sinks into the ground will be in the soil. And if it's in the soil, then a tree could bring that water up through the roots. As it moves up through the roots, it goes through the trunk and the stems, and when it gets to the leaves, it's going to be released by transpiration. So we've done all of these steps when we learned about the water cycle. None of this is new. What we didn't really talk about was transpiration and how it actually works. So, with transpiration in a plant, you've got roots, shoots, and leaves. Now, as it's growing older, you might have a root and a trunk and stems, and then at the end you have the leaves. In the ground, when there's water, the little molecules are hanging out there, and the roots are going to take water up through the xylem, the little hollow tubes that are made of dead cells, and it goes all the way out into all parts of the plant. Then you have other little tubes called phloem, and the sugars that are made through photosynthesis are going to move down and around to all parts of the plant. So the water is delivered to all the cells and the sugar is delivered to all of the cells. Transpiration is just the movement of water through a plant. It's going to move in through the roots, up through the rest of the plant, being delivered to every cell as it goes. And then when it gets to the leaves, there's openings in the leaves that we're going to learn about in the next video. And it leaves through the leaves. It's carried out from the leaves and it evaporates into the air. And that is transpiration. Need. I knew that was going to happen.
Want me to cut it?
No,